another interview of the series of interviews conducted by the Department of Chemistry, University of Colombo. I am Dimaki.
I'm Inoshi.
I'm Vishma.
We are undergraduates of the University of Colombo and we will be interviewing Dr. Dilhan Weeraratne. Dr. Dilhan is an expert in cancer genetics and precision medicine who currently serves as the genomics expert for IBM Watson Health. He has obtained his PhD in Neuroscience and Molecular Biology from University of Vermont in 2007. Subsequently, he has joined Harvard Medical School and Broad Institute, MIT, to do postdoctoral research in Meduloblastoma genomics. His work was pivotal in the development of the WHO-accepted molecular subgrouping of this pediatric tumor type, which was published in the journal Nature. He then transitioned to the biopharmaceutical industry, specializing in next-generation sequencing, cancer diagnostic, and targeted therapy. He has worked in several companies that are global leaders in precision healthcare, including Thermo Fisher, QIAGEN, and IBM Watson Health. Handing over to Vishma.
Thank you, Dimaki. Good evening, Dr. Weeraratne. Hope you are staying safe and healthy. And thank you very much for giving us your valuable time to take part in this interview. To start off with our interview,
Yeah, I just wanted to say that thank you very much for the opportunity to talk with you guys. It's always refreshing to talk with undergraduate students, which brings back memories when I was one many years ago.
So, Dr. Dilhan, to start off with our interview, I would like to know something about your background, like your early education and what made you interested in a field like genomics.
Okay, perfect. No, that's a great question. So, I come from a healthcare background. Both my mother and father were physicians, but so they were encouraged. So I always grew up with always hearing about medicine and treating patients. But at the same time, I kind of wanted to do something different from what my parents did. And when I was considering undergrad in the early 90s, the catchphrase or the buzzword was genetic engineering. And while I got selected to university in Sri Lanka, my first step in genetics took me to University of Delhi, where I specialized in genetic engineering as my undergrad degree. So, and so that was a good. And then from there, I proceeded to Singapore. I did my master's in genomics. I was using zebrafish as a model, and then in early 2000s. I came to the United States to do my doctoral work, and then I've been living here for the almost 20 years now, or 18, 19 years. So yeah, so given my upbringing, being very familiar with healthcare, that was what incentivized me to take a cutting edge, get into a cutting edge field like cancer genetics and cancer genomics. And then did my PhD in neuroscience, but soon after my PhD, for my postdoctoral work at Harvard and MIT, I transitioned to oncology, and ever since then, for the last 14 years, 15 years, I've been doing cancer genomics pretty much as my day job.
Doctor, as an Asian, what were the challenges that you faced when settling down in a country like USA?
So to be honest, I'll tell you, so I was always living in big cities like Colombo, New Delhi, then Singapore, each like huge metropolitan areas. And I also had lived in London as a child. When I came to the United States, it was almost a reverse culture shock from like big cities of like 3-4 million people. Suddenly I ended up in a place with like 25,000 people, small town, rural America. So that was like, for me, it was a reverse culture shock. And then I had, but in a very positive way. And also in terms of getting assimilated into the culture and society of the United States. I think the most important part is if you are good at what you do, there will automatically be recognition. So if you excel at whatever you do, that will create you many opportunities, and both in the social field as well as in your career. So my recommendation is that do the best what you can, and the process of integrating into the mainstream workforce is very easy. There is always good recognition when you excel at something. And also, yeah, go ahead.
No, thank you very much for the great advices.
So Dr. Weeraratne, when was your very first significant research work published, and what was it about?
Something I'm very proud of was when I was working at Harvard and MIT. I was, as you mentioned in your introduction, I was working on Meduloblastoma. Meduloblastoma is a pediatric brain tumor in the cerebellum of the brain, and usually this affects small children, typically ages 2 to 8 or 9, and it just like comes out of nowhere. So oftentimes I've seen that kids playing soccer or baseball here, and they complain of a chronic headache, and when they come to the physician, they get diagnosed with brain tumor. And as you can see, it's very traumatic, especially both for the kids as well as for the parents. So when I was doing this work back in between 2009 and 2012, all medulloblastoma patients were treated the same. So the basic treatment for this tumor type is whole brain radiation, aggressive cytotoxic chemotherapy, and also surgery. But what happens is when you treat little children with chemotherapy, and especially this is to the brain and the central nervous system, you get a cognitive impairment. So a lot of times we have seen that kids survive, but even if the kids survive the disease, their cognitive functions are severely impaired. So one of the things, like I used to tell in my talks when I was a postdoc, is that even if the kids survive, when the kids get to like 2021, that's like most of the time they are not independent. They live in their parents' basement, so the quality of life is impaired. The work we did was we sequenced about 200 pediatric tumors from Harvard Medical School and other areas, and we looked at the genomic profile of these tumors. And what we found was when we did whole exome sequencing, we found that there are actually 4 distinct molecular subgroups of the tumor. So what that means is, while the anatomical location of the tumor was the same, cerebellum of the brain, the genomic profile was very different. And then we did the survival analysis, and we found that there were 4 distinct subgroups, and the 5-year survival of these kids in these 4 subgroups are very different. So there was a subgroup which accounts for about 30% of the tumors which had activation of the Wnt beta-catenin pathway. Those kids, 99% of the kids survived. Whereas there was another molecular subgroup which was driven by amplification of a gene called MYC. Only about 25% of the kids survived in that subgroup. So what we advocated was, in that subgroup that had 99% survival even after 5 or 10 years, that these kids did not need radiation. These kids, and also very, very less cytotoxic chemotherapy. So so that's what we recommended to the WHO. Like there was a consortium of researchers, including myself and few other researchers. There was a group in Germany, there was a group in Canada. Now the standard practice is for this subgroup, they don't give radiation, so but only chemotherapy. And when you don't give radiation, their neurocognitive functions is much better. So their developing brain is not affected. So so there's a paradigm shift on how you treat this cancer as well as other cancers, that you basically don't give, like, you don't throw the kitchen sink at like any tumor. You just like tailor the treatment based on the molecular etiology of the tumor. But then again, on the other side of the coin, the subgroup that had very poor survival, even if you give radiation, their outcome is not better. So it's just that now we know in this brain tumor type, as well as in other cancer types, to stratify treatment, or like you tailor the treatment based on that patient's tumor. So something what we always tell in the field is that every tumor is very different. So even if you take two lung cancers, it will be dramatically different.
So, during your research work, have you ever had come across outcomes that were not expected? Can you share some of those experiences with us?
Yeah, so I think one of the most salient points is like just to one of the occasions, to get back into the example I told you about Meduloblastoma treatment. We always or the physicians always wondered, even in the United States, why some kids respond better and why some of the other kids do not respond. And then all the while we had a hunch that it might have to be something with the genetics of the tumor. But I'm talking about 2009, 2008. Human Genome Project was just completed in the United States, and tumor sequencing was not widespread at all. So all the cancer researchers in leading institutes were sequencing their favorite tumor type, and my favorite tumor type at that time was medulloblastoma. So it was incredibly gratifying to see that the clinical outcome was indelibly tied to the genetic makeup of the tumor. So while I wouldn't say it was, so at that point we know what to expect. This was an unexpected results in a very, very nice way, that it just confirmed our theory that that treatment outcome is tied to molecular etiology.
That's great.
To move on to our next question, how has genomics impacted other field of biology besides medical research?
So again, in medical research, you know that there's a huge impact on cancer, diabetes, other hereditary diseases, so on and so forth. Outside of the medical field, I think it has impact on multiple other fields as well. One of the things I can think of the top of my head is forensics. So the DNA footprint of a person can be inherently tied to forensic investigations. Another field that would have outside of the clinical field would be probably in law, especially it can, when you do genetic testing, it can either confirm your culpability or it can confirm your innocence. So as you may have heard, it's been used extensively in crime scenes, and it's a invaluable weapon in exonerating a person from a crime, or just tying or just confirming that the patient that the person has committed a crime. Another thing I would think is also in something like agrogenomics in agriculture. There are a lot of strains in like you get the beneficial genes for the crops. You make a hybrid thing where where the crops will be more resistant to environmental challenges, and the yield would be high. So there are so, and also in making genetically engineered farm animals to increase production and increase yield. So genomics is very widely used in agriculture, and also in terms of things like sustainable energy. So there are a lot of groups that's trying to create new energy sources based on microorganisms. So that's another emerging field that genomics will have a huge impact in time to come.
Doctor, can you share some knowledge about the current situation of the studies that are carried out in the fields of cancer diagnostic and targeted therapy?
Okay. You guys will have to stop me because I can go on for the next 1 hour on this. So tell me when you want me to stop, but I can give you at a very high level, right? So the first so basically what's targeted therapy? So you look at the basic, the conventional historical way to treat a cancer is that you give chemotherapy, right? So when you give chemotherapy, you not only attack the cancer cells, but you can't differentiate the tumor cell from a normal cell. So that's why when you take chemotherapy, you get like telltale side effects, like you lose the hair and stuff like that, because it pretty much target every single cell in the body, and which is not desirable. And also there's a very high incidence of recurrence. What targeted therapy does is when you sequence the tumor, you specifically target the mutation that's only in the cancer. So that way, when you give that drug through intravenous injection or even as a pill, it will target that specific mutation in the tumor cell and kill that tumor cell. So the first targeted therapy that was ever invented or just first approved by the FDA was a drug called imatinib for acute myeloid leukemia. This was back in like 2000. And then then the next progress was made for lung cancer. So one of the most frequently mutated genes in lung cancer is a gene called eGFR, and there were specific mutations in this particular gene. And there was a drug called erlotinib that was approved in 2004. And what that does is it specifically targets the lung cancer cell harboring these mutations, and that's why we call it targeted therapy. It only kills the cell with that particular mutation. And so till about 2010, I would say there were only 3 or 4 targeted therapies for cancer. But over the last 10 years, it has been exponentially increasing. So that now, at least I would say there's about 40 to 50 different targeted therapies targeting probably specific alterations in 25 to 30 different genes. So yeah, you guys feel free to interrupt me and ask any questions while we speak. If I'm talking too much at a higher level, let me know. Or if you want more explanations on what I tell, I'll be happy to provide them. So one of the classical cases was for melanoma back in 2012. There's a mutation called BRAF, mutation in a gene called BRAF, called V600E. So these mutations are basically in your nucleotide sequence. One of the bases get changed to another one, and because of that, the protein activity of that particular gene increases, gets augmented, which gives rise to uncontrolled cell proliferation. That's why you basically develop cancer. So you would have heard hereditary cancer versus somatic cancer, but in terms of most of the cancers, I would say 90 to 95% are somatic. So you are not inheriting this cancer. It's because you acquire that mutation or that cancer causing alteration during your lifetime. One example is if you smoke, you are more predisposed to getting something like lung cancer, or if you have exposure to something like asbestos, then you are more susceptible to getting mesothelioma. So, but then on the other side, you also get inherited cancers, which is much rarer in comparison to somatic cancers. And you guys probably already know this. Breast cancer, you can get breast cancer if you have inherited mutations in a gene called BRCA1 or BRCA2. So essentially, these inherited mutations, you get in every cell of your body, but it just predisposes you to getting cancer more. Whereas somatic mutations, you only get in the cancer cell, and so it's little harder to treat them inherited mutations with targeted therapy because you'll be targeting every cell of the body. But somatic mutations, you can hone in on the cancer and just treat the cancer. And one last thing I want to say is that I think last year there probably would have been 8 to 10 new targeted therapies that were approved by the US FDA, and some of the targeted therapy was the first time in certain cancer types. For example, in pancreatic cancer, there was a new drug. There was a drug called olaparib, first targeted therapy ever in pancreatic cancer. And then also in bladder cancer, for the first time in bladder cancer, there was a targeted therapy approved for mutations in a gene called FGFR. And also in cholangiocarcinoma, this is the cancer of the bile duct, which is a rare cancer but still very prevalent. They were able to identify a specific fusion only in that cancer type, and a drug was approved earlier this year. So precision oncology is taking huge strides. Almost for every cancer type, we have identified one or two alterations, and then concomitant drugs that will target those specific mutations. Any clarifications? Oh. Okay, yeah, go ahead.
Dr. Weeraratne, I heard that you are taking part in projects related to personalized medicine. In a time where there's no exact treatment for cancer, how can we apply personalized treatment for cancer patients, and also how practical it is to implement them in the common society?
So I think, so personalized treatment, you can think, the way I look at it is like, so what I spoke about just now, targeted therapy, in a way, is actually personalized therapy. Like I said, every cancer is different, right? So even in the US, what usually happens is when somebody first gets cancer, there are what we call standard of care. And then, so while there are many targeted cancer therapies that are approved in the US now, but usually the first line of therapy is probably surgery or radiation or even standard chemotherapy. But as you know, what happens is, say if you have a breast cancer, then you get tamoxifen or like standard of care. Then the cancer may come back, especially things like colon cancer. And when you come back, then you need to, and then the tumor metastasize. So metastasize is like if the cancer originates, say, in your colon, after few rounds of treatment, if it is very aggressive, it can get into your liver, it can get into your kidneys, it can get into your peritoneal cavity, and it can spread. So at that point, standard care will not work. So that's when we advocate here in the US that you need to sequence your tumor and see what kind of mutations are there. Then you can personalize treatment for your cancer. So you ask how practical it is, right? So that's an excellent question. So a lot of this new targeted therapy, if you don't have insurance, it's very expensive. So some of those drugs I mentioned for lung cancer, erlotinib or gefitinib, if you are paying out of pocket, it's about 15,000 US dollars a month. So I mean, no one, if you don't have insurance, there's no way that you can afford it. But then that brings to the question that in less privileged areas in the United States, or if you don't have if you don't have access to health insurance, or if you are underinsured, how can these patients patients afford? And then that brings to places like Sri Lanka or any other developing country where, to the best of my knowledge, most of these drugs are not available. Even, I mean, most of the drugs are new even in the US, but especially because they are very extremely expensive, and there are no generic drugs yet, because these pharmaceutical companies are making a killing out of that. So like I said, it's $15,000 a month, so it's roughly 200,000 just for one patient. But and they can do that because till their patent expires, right? So once their patent expires, then you can make generic drugs. But till that time, I think one of the main barriers trying to implement this in society is the cost. And also a lot of oncologists, you might find it little hard to believe, but I've seen in the United States, there are a lot of oncologists in community practices who still really don't know at what point that they need to give targeted therapy. Even physicians, they are resistant to change. So they would like to practice what was taught in their medical textbooks like 20 years back. Things are changing here now, especially in the bigger cities, but still we find that that interpreting next generation sequencing results can be a challenge for physicians and oncologists unless you are specifically trained in that field. Yeah, so those are the challenges in implementing personalized medicine for cancer, but things are getting better. And hopefully it'll be targeted therapies will be available in countries like Sri Lanka in the not so far away future.
All right. Dr. Weeraratne, computers have changed the way that genomics research investigations are conducted. So we would like to know how artificial intelligence has been used to improve the quality of genomic studies.
Yeah, so I can give you two examples. So one of the, especially for, so I've been talking about mutations in different types of cancers, right? So one of the biggest challenges is that you need, so some of these mutations are pretty rare. So there's so that particular mutation I mentioned in melanoma, BRAF, this mutation is also present in thyroid cancer, but the prevalence is very low. So you need to sequence thousands of patients in order to identify a patient with that particular mutation. So you might think that if there are 50,000 patients with a particular mutation in a particular subtype, and if there are only 30 patients with that, um, why do that? But then this is the beauty of precision medicine, but that will help those 30 patients to recover from the cancer. So one of the ways AI and machine learning has helped in genomics research is that when you sequence the entire genome of the patient, so you know that if you do whole genome sequencing, it's about 3 billion base pair. If you do whole exome sequencing, that's 30,000 genes. So it's manually very hard to analyze and analyze such amount of big data. So there are so many new algorithms to pass that all raw sequencing data and there are machine learning algorithms to subtract the noise from the signal and to identify the actual mutation in that patient's tumor. So this is something that would have been do without computational algorithms. The second way machine learning and artificial intelligence is helping is for the clinical interpretation of this variant. And just by going into PubMed, how many new papers are published every day, right? So there's probably, I can't remember off the top of my head, at least 500,000 papers every month on different aspects of cancer genomics. So some of these papers are probably not reliable, but then it's humanly impossible to pass through all this new published data to accurately interpret. So there are a lot of machine learning algorithms that helps the analysts and the scientists to pass through this data just to identify the relevant information. And I know at IBM Watson Health and other places, there are multiple machine learning and natural language processing algorithms that help scientists to go through these copious amounts of data to identify clinically relevant information. So that's two of the tangible areas that AI and machine learning has played a big part.
Nowadays, computers have started beating humans in every field. Will doctors be replaced by computers in the future? What is your idea about it?
That's an excellent question, question, Inoshi. So the way I look at it is, so this is how I would look at it. There are certain things that humans excel, right? So humans are very good at empathy, compassion, dreaming. There are certain things that machine can't do. Also things like, especially morals, compassion, But then on the other hand, there are certain things that machines do better. Things like pattern recognition, reduction of bias, or things like, increasing scale. So scalability is a big problem for humans, right? So the way we look at it is that artificial intelligence will not be able to replace physicians in the near term or even in the longer term. But what I call is, it's not artificial intelligence. I would like to call it augmented intelligence. So we take advantage of things that the machines do better, and then combine that with things we humans do better. So augmented intelligence is something that in the field of medicine as well as in other fields that I think people should definitely make use of it. So you asked from me whether artificial intelligence will ever replace humans. But my answer to that is physicians who are not using artificial intelligence will be replaced by physicians who are using artificial intelligence. But you do have to use AI-based things, and all these things help them to identify and treat better, and not only in genomics, in things like radiology. So there are so many new algorithms that would help you to identify lesions when the human naked eye won't be able to recognize this. In digital pathology, you rather than a normal hematoxylin slide, there are new algorithms where you can just look at the pathology slide and then identify and stage the disease. So it is imperative that physicians need to embrace AI and machine learning. But as you said, one of the inherent human fears is that doctors are not going to have jobs in 20 years, but that's not going to happen.
So, apart from cancer therapy, what are the other important projects that you have been working on recently?
Oh, yeah, thank you for that question. So I was doing active, lab research and active academic research till 2013, when I decided to leave academia and get into the corporate world. But over the last 2 years, I have gotten back a bit into primary research, and with the advent of COVID pandemic, pretty much all organizations, made a reactive decision to work on the COVID-19 pandemic. So at the beginning of the year, myself and a few other folks at IBM started working on COVID genomics. And I'm not sure whether you have heard of the mutation, the COVID-19 mutation D614G. So this particular mutation was not prevalent in the strains in China. But in March and April, myself and 3 other folks from IBM were the first people in the world to report this particular mutation. And if you could go to the PubMed, that would be the first publication. And after that, there were probably 200 different publications on this particular mutation. And this has become the most prevalent strain right now in the US as well as other parts of the world. But it's present probably in 80 to 90% of COVID-19 strains. However, in the original Chinese cohort, it was probably present in 1% or less. So in that publication back in March, end of March, we said that people who are developing vaccines for COVID need to look at the mutational spectrum of different COVID strains, because one vaccine may not fit all, right? And now you may have seen the new news last month that there's a different mutation in the UK that people are worried that Pfizer vaccine and the Moderna vaccine may not work. So the same concept. So whatever we had reported back in April has become the dominant strain. So, and the vaccines were developed against that. But when there's a new vaccine, there's always a question: would the vaccine work? In my opinion, it should, but still, that's something that you would need to experimentally validate, right? So that's one area that I'm working on. In addition to so besides cancer work, and then we have also developed application where you can visualize the spatiotemporal genetic variations of COVID-19 strains from beginning of February till now, how there are substrains which gets confined into different geographical regions. Yeah.
You have been fortunate enough to work at the world-renowned Harvard Medical School Broad Institute, MIT. So we would like to know how you made your path to get there.
Yeah, that's a great question. I think there are a couple of things. So if you have a passion on something, definitely develop that. Do a lot of networking, and 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 then have a clear mind, clear goal. If you are doing a doctoral study wherever in the world, or even undergraduate studies, that if you know that this is what you want to do within the next 5 years, then have a clear path uh, on, uh, on getting there. So in my case, I did neuroscience for my PhD, and I was working on mouse models, and believe it or not, I did a lot of work with paramecium. So I did a lot of paramecium genomics, but still I wanted to move into human clinical research, and directly after my graduate studies, I went to multiple interviews and got multiple positions in different cancer labs at Harvard and MIT. But it was just that I did not have cancer experience. I had experience in paramecium genomics, but it's that if you have passion for something, you will be able to convince that your skills will be translatable to a different discipline.
You have been working in cross-functional teams, including software developers, bioinformaticians, and oncologists. So what sort of experiences have you gained through such interactions?
That's an excellent question. So as somebody who has worked in academia as well as in the corporate world, one thing, so in academia, I've seen that people can work in silos. You do what you do best, and then somebody else is doing what they do best, right? But probably things are changing a little bit in academia, but especially in the corporate world, you need to interact with people with different backgrounds, different disciplines. So oftentimes when I work with software engineers, they have zero background in biology, zero background in sometimes even in science. Right? So I think the biggest thing I've learned is to communicate science to somebody who is not a biologist or who's not a physician. So when I talk to oncologists, I have you would need to learn to talk at their level, whereas when you talk to somebody like a computer programmer, then you need to speak their language basically in order to make it very clear that what you want to get done. So the ability to work in a multidisciplinary environment is something, a very valuable skill set. I think a lot of people should have.
My next question for you is, joining a company like IBM Watson is a big achievement for a scientist. So can you share some of your experiences and the research work that you are doing at IBM Watson?
Sure. Yeah. So again, I joined IBM Watson about 2.5 years now. I was working as their global cancer genomics expert, but then lot of times in companies, what I come to realize is, unlike in academia, the business strategy changes, business priorities changes. So you have to be very open-minded to doing something today. When the business strategy of the organization changes, you would need to align with the new priorities. So like I said, when COVID-19 pandemic happened, 90% of my work was related to COVID-19, but not so much anymore, because things are the things are under a little bit control right now, so to speak. But so the and another thing, this is one of the things I'm really getting interested in, is at IBM, I'm working on health equity. Health equity is that not everyone in the world has access to same type of healthcare, right? So this is a segment of equality, or because we've seen a lot of disparities in healthcare in the United States, and everywhere in the world, even in Sri Lanka. But that's something from a social demographic point of view which has been neglected, and I think it's a basic human right that everyone in the world should have access to healthcare. But for a lot of so been doing a lot of research on health disparities, and especially Hispanic population in the U.S. and African American population in the U.S. don't has they get different healthcare for variety of reasons than say for Caucasians. So it's imperative that that there should be equity in healthcare, and to break down barriers that prevent people from accessing healthcare in a similar way. So that's a lot of companies are focusing on that to as a bigger part of the disparity problem in the world.
That's really great, I think. So you work as an advisor for physicians and oncologists on the use of tumor biomarkers. Can you please explain a little bit on how these tumor markers are made use for treating cancer patients and their importance in cancer treatments?
Yeah, I think we spoke about this extensively at the like little while back. This is basically you sequence a tumor, and then you find a mutation, and then your cancer treatment is based on that particular mutation. So, like I said, so there's generally like first line of treatment is when you first get diagnosed with cancer, and at that point cancer is localized, meaning that if it is in the breast, it's still in the breast, and you can just treat with chemotherapy or mastectomy, like removing the surgery. But when the cancer gets to an advanced stage, say stage 3, stage 4, the cancer spreads to other parts, your regular treatment like chemotherapy and radiation may not be sufficient or will not be sufficient. So at that point, that's where people like us, myself, come in, that we sequence the tumor and then advocate to the treating oncologist that this patient has this so and so mutation, and there's this so and so newly FDA approved drug. You should treat your patient with that. Or on another level, a lot of patients, when patients have mutations, they're eligible for clinical trials that's treating patients with that particular mutation. So these are not FDA approved, but clinical trials are essential for approval of drugs, right? So when we identify a mutation in a patient, then we would encourage the physician to enroll that patient in the clinical trial. So there may be durable clinical benefit to the patient, and that will also help advance the clinical trial. So this happens when there's no other feasible treatment for the patient. So the cancer has spread to other places, and clinical best course of clinical action.
So in your point of view, what is the scope of a genomic researcher in relation to upcoming trends in the world?
So I'm going to go off in a limb and say that probably in 20 years' time, if you have a disease or even at birth, you probably will be, your genome will be sequenced. And that would give direction to what you should be doing. And even now, if you have predisposition to cancer, say there are certain people who get colon cancer at the age of 30. The average age for colon cancer is like 65 to 60, but we still see we see few males getting colorectal cancer at the age of 30 or 35, and in many of these cases, that's because the patient has inherited a bad gene from the parent. And so if a male gets colorectal cancer at the age of 35, or endometrial cancer, a female gets endometrial cancer at the age of 30 or 35, then it's if they have children, it's very important that that you monitor them. So if your father had cancer at an early age, you should go for genetic counseling. You should go for genetic screening periodically, so that will, while it won't mitigate your chances of getting, you can detect the cancer early. So so I think, as you said, the scope of genomics or genetics, I think we are moving to a world of genomics-driven care. So we see that very palpably in cancer right now, but even in other disease areas like diabetes, then then cardiovascular diseases, there's a genetic component to it. And as our knowledge grows in the coming years, I think healthcare will be driven more and more by genetics and genomics. So that's one of the, especially, and we all know that in healthcare, this is going to play an important role. And as we discussed earlier, genomics is also affecting other areas like law, forensic, agriculture, and so on and so forth. So I guess sky is the limit.
COVID-19 pandemic has been a major global issue in the past few months. So how did this affect your work, and how did you face those challenges that came with the pandemic as a scientist?
Well, especially here in the United States, you probably know pandemic was not controlled well. so so most of the time we have been working from home, and they're in, like, so there are ups and downs. Obviously you don't get to physically see people and work together, but at the same time there are a lot of video calls, and it's just getting adjusted to working remotely. But I think people have accepted that challenge and embraced the difficulties of working remotely. So that's what I would want to say.
A review of emerging therapeutics of COVID-19 has been published with your contribution. This has mainly focused on the efficacy of emerging therapeutics for the use in treating COVID-19 and also identifying the best available treatment options to deal with the current pandemic situation. What do you think about the emerging RNA vaccines?
I think almost every scientist was surprised at the initial efficacy of these vaccines. As you know, Moderna has been trying for RNA cancer years, but obviously getting a cancer vaccine is harder than a vaccine for a virus. But regardless, the Pfizer vaccine had 94% initial efficacy, and the Moderna 95%. So it's incredible. If you had asked 1,000 scientists in March whether this would work this way, I don't think anybody would have expected this kind of efficacy. Even initially in June, FDA was going to give approval even at 50%. This is incredible, like over 90%. But again, my only slight concern is that this is the first time that RNA vaccine has ever used or has been approved for humans. We don't yet know the long-term effects of manipulating your RNA with a exogenous RNA, right? So most likely will be safe, but we are yet to see the very long-term effect, say 2-year, 3-year effects. But that being said, there are other companies like Johnson and Johnson and Novavax. Their vaccines should be approved hopefully within the next month or two, and they are not RNA vaccines. They are your typical vaccine, which is created against the spike protein of the COVID-19. So they are more of your standard vaccine. And my other slight concern is that storage of this RNA vaccines. The Pfizer vaccine should be stored at -80 Fahrenheit, whereas the Moderna is -8 Fahrenheit. And especially in the supply chain, I worry, I wouldn't say worry, something you need to think is that you need to make sure that the vaccines are stored at -80. Otherwise its efficacy won't be the same. Now, your traditional vaccines, like what Johnson and Johnson and Novavax are developing, you can store those vaccines as like even in a fridge at 4 degrees. So that way, long term for global distribution, the traditional vaccines may be more pragmatic than an RNA vaccine, perhaps only for the reason of global distribution at -18.
Doctor, do you think the trial data that researchers have are sufficient to give that vaccine to the entire population of the world?
Yeah, again, yeah, this brings to my previous answer. Like I said, the efficacy is phenomenal, 95% efficacy. And so again, you guys, I'm sure you guys are familiar with, at least peripherally familiar with the data. So it's that I think Pfizer, so usually they give the placebo to one arm, that's saline solution, and then the vaccine to another segment of the trial population. I think in Pfizer's and Moderna's case, like 15,000 patients got the vaccine, and 15,000 patients got the saline. But out of patients who got COVID, I think maybe about 150 who got the saline got the disease, whereas only 10 that got the vaccine got COVID-19. So that's how they calculated the 95% efficacy. So I think since 30,000 people, it's a big number, and still they are doing phase 3 trials. So these results are very impressive. But again, my only concern is the long-term side effects. So which I don't foresee, but still we need to clinically validate of mRNA vaccine.
All right. Doctor, you are residing in a country that possesses cutting-edge technologies. So, in your opinion, what fields of chemistry will have most demand in the future? What will be the future of chemistry and research?
Okay. Let me try to answer this as well as I can, because I'm not a chemist, but I have a lot of friends who are in chemistry and who are working in pharmaceutical companies. I think one of the areas would be nanoparticle chemistry. One of the things scientists are working and have been working for a long time is drug delivery, nanoparticle vesicles, which could directly go into the tumor cell or the any disease cell. So that is an area I think that will have a lot of scope and potential in the near term. And also, I would say chem-informatics. So that is the marriage between your traditional chemistry and informatics. And informatics is, as you know, is impinging into almost every areas of science. And again, I think we should embrace computational knowledge in order to support your findings, be it in biology or be it in chemistry or physics. So those yeah, and also medicinal chemistry as a broad field is having a tremendous impact in drug development, drug and vaccine development. So that's a field that's very much in high demand in the United States and in other countries.
Dr. Weeraratne, I was asking, would you like to tell us what you like to do when you aren't working on research and any other fields apart from research and academic work that you might have interests on?
Yeah, you guys probably didn't hear my answer because we are going in and out. So what I said was, so instead of so in addition to cancer, I've been working, especially this year, I've been working on other areas like COVID-19, other chronic diseases like hypertension, diabetes, and then also health equity. So in addition to what I do for a living, I love traveling. I love hiking, and I especially love cooking. And now also I have two young kids below the age of 10, so that keeps me occupied and busy most of the time. So that's a pretty, yeah, that's a handful.
So before wrapping things up, I would like to ask a very important question from you. As a scientist, what advice would you like to give to students who are interested in science and who are planning to enter the field of science research?
Yeah, so one of the things I want to emphasize is, so you might probably, so if you are thinking of doing graduate studies, you might probably have an idea what you wanna do. My advice is when you enter or when you enter a lab as a scientist, or when you start doing research, do a bunch of things. See what you really want to do. So especially in the United States, when you get into a graduate program, you have what you call rotations, that you spend your first year working in 3 or 4 different labs. That way, you know what you really want to specialize in, or what you want to do, and then you get exposed to different areas of research in your field. Once you identify an area which you are really passionate about, then you know that's make a name for yourself, give presentations, make yourself the person in that field. So it can be a small sub. If you are into academic research, it doesn't need to be a very broad area. You could be the world's expert in a small niche field, but the challenge or what people need to do is to identify that area where you want to be the world's expert, or like one of the preeminent people working on that area. So other thing is special. You can work in a reputed lab, which is good and bad. There'll be it may be hard if there are people in that particular area. It might be able. It may be difficult to establish your position in a crowded area. But that being said, if you work in a famous lab, you still carry that reputation. And especially as a graduate student and a postdoctoral researcher, you know this, your currency is publications. So if you get a lot of first order publications, first and second order publications, that will take you a long way in your career. The other advice is, when you know what you want to work on, do make your know, talk to people, go to a lot of meetings, a lot of conferences, present your work. So that will help you to establish your reputation in the field.
Thank you again, Dr. Weeraratne, for allocating your valuable time for us. This interview has really inspired us to see new dimensions of science. So to wrap things up, today we have interviewed Dr. Dilhan Weeraratne, who serves as a genomics expert for IBM Watson Health. Hope you all enjoyed the session. Thank you, and stay tuned for an interview of another scientist. Have a nice day.
Thank you very much. It's my pleasure to talk with you guys.